Hey everyone, welcome to my video series on Nextflow and Vultr. In this series, I'll show you how to run a real bioinformatics pipeline in the cloud using real sequencing data. Let's get started. So what is Nextflow? Nextflow is a workflow management system commonly used in bioinformatics, and it makes it possible to run reproducible and scalable analysis pipelines across a wide range of computing environments, from laptops, university clusters, to cloud servers. And one of the things I like most about Nextflow is the NF Core ecosystem. And NF Core is a community-driven collection of standardized bioinformatic pipelines that anyone can use for free. So whether you're working with RNA-seq, whole genome sequencing, metagenomics, or many other types of data, there's a good chance an NF Core pipeline already exists for your analysis. In fact, these pipelines are widely used throughout the bioinformatics community, and you'll often see them cited in the materials and methods sections of published studies. And for this tutorial series, we'll be working with a real RNA-seq dataset from a yeast experiment investigating the role of the transcription factor RAP1. And to keep the analysis manageable, we'll use a subset of samples from the study, but we'll follow the same workflow you would use for your own sequencing data. And each sample in this study contains approximately 100 million sequencing reads, making this a realistic example of the type of data researchers often work with. Of course, running these pipelines requires computing resources, especially for large sequencing data sets. And if you don't have access to institutional computing infrastructure, or if those resources are difficult to use, cloud computing can be a great alternative. And there are many cloud providers available, including Amazon Web Services, Google Cloud, and Alibaba Cloud. But in this series, we'll be using Vultr, because in my experience, it's affordable, easy to use, and quick to get started. So over the course of this video series, we'll learn how to launch a cloud server, install the required software dependencies, run a test analysis, upload and process real sequencing data, download those results back to our computer and inspect the results, and review costs and shut down the server. And if you'd like to follow along, feel free to create a Vultr account and download the dataset linked in the video description. Otherwise, sit back and enjoy the video series. I'll see you soon.